All right, now that we have some people here, I'll go ahead and get started. So good morning and thank you everybody for joining us for the virtual 2020 Minicon. For any of you that don't know me, my name is Casey and I'm the chair this year for the Minicon. It's been fun putting together a virtual version of it. While everyone's kind of getting settled in and we're letting other people join, I'd like to start off by quickly going over some logistics of how the Minicon is going to work this year, since it's different, how we're going to address questions, how you guys can ask questions, and just give you some information like that. So the first part is when we're switching between speakers and panelists, it might take a minute or two to get everybody switched over because each person is going to be switched from an attendee to a panelist, and then when they're done, it'll be back from a panelist to an attendee, just to kind of keep everything simple and not have any interruptions. And this is going to be done by the hardworking third years behind the scenes. Award recipients will also be changed from attendee to panelist as soon as they're announced. It's completely your choice if you want your camera on. You don't have to have your camera on. You can just have your microphone on if you want, but I'm going to promote you to a panelist so that you can be heard or seen if you choose to do so. The next big thing that I'm sure a lot of people would have questions about is how to ask questions. Since I have you guys all as attendees and I don't want to have, we have some logistical issues with being recorded. How we're going to address asking questions is if you have questions for the current panelist or the current person presenting, please type it in chat as soon as they're done. And we will have one third year that will be responsible for reading those questions to the presenter. And then when it comes to coffee breaks, there's going to be 2 scheduled coffee breaks to kind of allow people to get up and stretch. We'll have just a regular screen being shown that says coffee break on it. I wish I could give you guys the option to chat or mingle with each other, but it's not really easy to be done because I'd have to go through and switch everybody to be able to talk and then back off. Hopefully that covers the questions and how the logistics of how we're going to run the Minicon. So I will leave it there and I will switch it over to our wonderful MCs, Melissa and Rami.
Hello, everyone. Welcome to Minicon 2020. We're happy that you are able to join us today. Melissa and I will be your entertainment as your MCs. And just to let you know, since we are broadcasting from our home to yours, we appreciate your patience if there are any technical glitches or noise issues, and appreciate your flexibility as we adjust to this unique presentation where we have participants and attendees joining us from all over the world. To start things off, I'd like to introduce our esteemed chair, Professor Michael Miller.
Welcome. Am I on? I'm not sure if I'm on.
You are on.
Okay, great. Well, welcome to the 2020 Psychological Brain Sciences Minicon. This is always a really special event for us. This is something I know the faculty very much look forward to every year. It's great to see all the terrific work that our second year students have accomplished by this point. It's great to normally be able to see everybody, but we can't do that this year. But I think this year is particularly special. I think it was great for Casey and Melissa and Rami and all the other third year students to put this together. I think this is important to try to do the best that we can do to keep things going at this time. And just to be able to, keep that connection with everybody. I know a number of grad students weren't able to complete their work this year due to the shutdown, and they will hopefully be presenting their work at some point next year. But for those hardy souls that were able to pull through and put together a presentation, I think we all greatly appreciate it. So with that, I'd like to welcome you and kick off the Minicon. Back to you, Rami.
Are you seeing the screen share? Okay, great. So our first presenter is from Nancy Collins' Close Relationships Lab in the area of social psychology. She is from not too far away in Thousand Oaks, and here she is from a very young age wanting to tell you about all her research ideas. She completed her undergraduate education at UT Austin, and in her spare time, she is very active, having completed the Santa Barbara Sprint Triathlon. Please welcome Paige Harris.
Okay, sorry, let me share my screen. Okay. Can everybody see this? Yeah, okay. Go ahead. Good morning, everybody. Today I will be talking to you guys about some research that we've been doing about how physical activity is associated with positive intrapersonal processes and interpersonal outcomes. So I want to start off my talk today with this infographic from the World Health Organization. A major public health initiative at the WHO is to increase physical activity engagement. And as you can see here, the WHO recommends that adults engage in 150 minutes of moderate physical activity each week. And doing so provides a host of intrapersonal benefits. As many of you know, physical activity improves physical health, including promoting cardiovascular health and reducing stress reactivity. In addition, physical activity has been shown to improve both objective and subjective sleep measures, or subjective measures of sleep quality. Further, physical activity improves cognitive functioning in a variety of domains assessing executive control. And finally, physical activity improves overall mood and mental health in both clinical and general populations. In sum, we know a lot about the intrapersonal benefits of physical activity. Past research clearly shows that when people engage in activity, they sleep better, think more clearly, and are happier. So this made us wonder, could the benefits of physical activity extend beyond the individual? That is, could these personal benefits spill over into the social domain and help foster stronger social relationships? To date, no study has directly examined whether physical activity is linked to positive social outcomes, but this hypothesis was first proposed in the popular movie Legally Blonde. Here, Elle Woods is talking about her client, an exercise trainer who is accused of murdering her husband. She says, exercise gives you endorphins, endorphins make you happy, and happy people don't shoot their husbands. So as you can see, Elle proclaims that physical activity leads to positive physiological and mood benefits that then enable people to behave pro-socially. While we don't test her exact hypothesis, we do think that Elle is onto something. And this brings us to our theoretical model. As discussed earlier, we know that physical activity promotes positive mood, enhances cognition, and improves sleep, which captures this first piece of the model. We propose that these adaptive intrapersonal processes then lead to positive interpersonal outcomes, which captures the second half of the model here. Prior research shows that positive emotions, executive control, and good sleep can improve interpersonal behavior. Thus, we hypothesize that physical activity restores emotional, cognitive, and physiological resources that then enable people to behave more positively toward close others. These increases in pro-relationship behavior should then have downstream consequences on social relationships. And this brings us to the current study. The primary goal of this study was to examine the interpersonal benefits of physical activity and to provide an initial test of the model by exploring potential mediators. We wanted to assess these processes in a naturalistic way, so we designed a daily diary study in which we tracked people's physical activity and social behavior every day for 2 weeks. A diary study has a number of key strengths. Most importantly, it allows us to distinguish between within-person and between-person effects. The within-person effects allow us to see how people change from day to day. So this means that we can compare people to their own baseline and see how their social outcomes differ on days when they are more versus less physically active. The between-person effects allow us to compare the social outcomes of people who are more versus less physically active in general. So for this study, participants were 173 undergrads from the subject pool. They were mostly women and they were racially and ethnically diverse. Every day for 2 weeks, participants completed 2 electronic diaries, one when they woke up in the morning and one in the evening before bedtime. The morning diary asked questions about their sleep quality from the night before. The evening diary asked participants to report about their behavior and experiences that day. It included questions about how sharp and focused they felt, their positive emotions, their physical activity, and their social experiences that day. So participants answered a lot of questions, but I will briefly review the measures we used in the analyses for today. Daily physical activity was measured with one item that assessed overall physical activity that day. We also included daily stress as a control variable, which was assessed with one item. Based off validated scales, we measured daily positive emotions with 10 items, daily cognitive focus with 2 items that assessed feeling sharp and focused, and daily sleep quality with 5 items. For our interpersonal measures, we assessed daily responsiveness to close others with 4 items. Importantly, these items measured the extent to which the participant was being caring and understanding of others. And we assessed daily social connection with 4 items to get a sense of how positive their social interactions were and how connected they felt to their friends and family. This brings us to our key hypotheses, which focused on the within-person changes on all outcome measures. So we hypothesize that on days when participants are more physically active, they will experience more positive emotions, experience more cognitive focus, get better sleep that night, be more responsive to close others, and feel more socially connected to close others. Finally, we predicted that the links between physical activity and the interpersonal outcomes would be mediated by the intrapersonal benefits. Before we dive into the results, I want to briefly review the data analytics strategy. Given the nature of our data, we use multi-level modeling in SPSS and a macro to conduct the multi-level mediation analyses. In all analyses, we modeled the within-person effects controlling for the between-person effects. We controlled for the between-person effect to rule out individual differences in average activity levels. We also controlled for stress and day of the week. We controlled for stress because people may be more physically active and socially engaged on low stress days versus busy, high stress days. Likewise, we controlled for day of the week because people may be more physically active and socially connected on the weekends versus school days. First, we wanted to see if daily physical activity was associated with our daily positive intrapersonal processes. So we first tested if daily physical activity predicts daily positive emotions. And so first, I will orient you to the table here. This highlighted section is our within-person effect. So as you can see here, that daily physical activity did indeed predict daily positive emotions. And as shown on the graph, on days when participants were more physically active, they experienced more positive emotions. Next, we looked at cognitive focus that day. And we found a similar pattern of results. As you can see, on days when participants were more physically active, they reported feeling more sharp and focused. But what about the link between physical activity and and quality of sleep that night? For this, we conducted a lagged analysis in which physical activity predicted sleep quality reported the next morning. Unfortunately, we did not find a significant association. Physical activity during the day was unrelated to sleep quality that night. So, in summary, on days when participants were more physically active, they experienced more positive emotions and they felt more sharp and focused, but they did not sleep better that night. As such, the first set of findings are largely consistent with our theoretical model. The next set of analyses explore the association between physical activity and our social outcomes. We first wondered, would people behave more positively toward close others when they were more active? And we were excited to see that on days when people were more active, they reported being more caring and understanding of their friends and family. But what about our most distal outcome? Would daily physical activity be related to daily social connection? We were also really excited to see that when people were more active, they reported feeling more close, connected, and in sync with friends and family. So to sum it up, on days when participants were more active, they were more caring and understanding, and they felt closer and more in tune with their friends and family. This is the first diary study that we know of that associates physical activity with positive social outcomes. We were really encouraged by these findings. So finally, we ran multi-level mediation analyses to further test our model. We used positive emotions and cognitive focus as mediators because they were significantly related to physical activity. We first tested whether positive emotions mediated the paths from activity to our interpersonal outcomes. And we found evidence for significant mediation. When people were more physically active, they experienced more positive emotions, and these positive emotions then predicted more responsive behavior and more social connection. But what about cognitive focus? Would feeling sharp and focused mediate the association between activity and our social outcomes? And in this case, we found that cognitive focus significantly mediated the link between activity and social connection, and it marginally mediated the link between activity and responsive behavior. So on days when people were more active, they felt more sharp and focused, and having this cognitive clarity then predicted better social outcomes. Overall, the findings are encouraging. On days when people were more physically active, they felt better, their thinking was clear, they were more caring and understanding, and they felt more close and connected to close others. Importantly, we found evidence that the positive intrapersonal processes explain the positive social outcomes. And consequently, this study replicates prior work showing that activity is related to personal benefits. And it is the first to suggest that activity may also have really important benefits for our social relationships. Although we are really excited about these findings, it is important to recognize some limitations. Our sample included young and healthy college students, and our findings are based on self-reports. And because our study is correlational, we cannot draw any causal inferences. Therefore, in future studies, we plan to extend our work in two important ways. First, we plan to conduct another diary study using activity monitors to provide objective assessments of activity each day. Second, we plan to conduct experiments in which we manipulate activity in the lab and test causal impacts on social behavior. And with that, I'd like to thank you guys and everyone who's made this possible, my wonderful advisor, Vicky, and my lab groups, and my research assistants. Thank you. I'm gonna stop sharing now. Do I stop?
All right, Paige, can you hear me okay?
Yes.
We have some questions for you in the chat. First of all, great job. And so from Dr. Miller, we have, how much of a role does the physiological response play on social connection versus just the satisfaction of doing physical activity?
That's a great question. I wish I knew the answer to that. I definitely think that the everything that's going on physiologically has a huge role, like what's going on inside the brain, and then what's happening in your body. I think that plays a huge role in how we are, got distracted, sorry, and how we feel, and then how we also like engage with our close others. I don't know how much, if it plays a bigger role than feeling satisfied about actually doing the activity. These are all things that I've talked about with my lab groups. So these are things I definitely want to explore, but I don't know, how much of it is physio versus the satisfaction of doing it. Thank you for that question, though.
Great. Our next question is from Chris McFerrin. Was diet assessed at all?
Thanks, Chris. No, actually, we did not assess diet. That would be really interesting, but no, we actually, we did assess, like we had a few questions about self-regulatory behaviors, like did you eat too much today? Did you think that you ate too much today, or did you think that you ate too little? Did you not eat enough? And they would just check that. So that's kind of the only assessment we have of eating and food. But that would be interesting to see. We didn't ask it because we had way too many questions in this diary study, but maybe for future ones we will.
Quickly, Paige, before you finish up, because I think there's no more questions. Are you and Vicky on the same computer again, just for the panelists that have to promote you? So the panelist that promotes Vicky, just keep in mind that she's Paige. All right, I just wanted to make sure. Thank you. All right, we can hand it back over to the MCs.
Okay, give me 1 minute. Can everyone see my screen?
Are we presenting? Okay.
Yes, I'm just trying to share my screen. Can you see my screen? Okay, great job, Paige. Our next speaker comes from Richard Mayer's lab in CPCN. She is a Santa Barbara native who recently won a National Defense Science and Engineering Graduate Fellowship. Not only has she already published 2 papers since starting graduate school, but she recently adopted a dog. Here to talk about generative learning strategies, Alyssa Lawson.
Okay, let me share my screen. All right, can everyone see my screen? I'm going to assume that's a yes. So, hello everyone. Before I start my talk today, I want you to imagine a scenario. So imagine that you're trying to learn about greenhouse gases and you find a lesson online. You're going through the lesson online, however, you feel like you're not really learning any new information. What can you do to better learn during this lesson? That's what I'm going to talk to you about today. So first, I'm going to start with generative learning theory. And this is a theory that states that students participate in deep learning when they engage in appropriate cognitive processes. These cognitive processes are selecting the relevant information, organizing it into a coherent structure, and integrating it with prior knowledge. And since deep learning is a goal in education, we need to think of strategies that prime these processes in order to get students to engage in deep learning. And a way to do this is to use generative learning strategies, which are activities that require learners to engage more deeply with the material by priming those 3 cognitive processes. When that's done, that improves learning in the learners. And there's a lot of different types of generative learning strategies. However, the one that I'm going to focus on in this talk today is called self-explanation. Self-explanation is the activity that requires learners to explain the material in their own words while they're learning. And there's been a lot of research looking at self-explanation and the benefits of using self-explanation while learning. However, one of the boundary conditions to self-explanation is that sometimes students have difficulty making explanations. And some reason why is that self-explanation isn't just one thing. It's actually a continuum. There's a lot of different types of self-explanation that vary in the level of assistance that's provided to the learner in creating their explanation. On one end, we have no assistance provided to the learner, and on the other end, we have a lot of assistance provided to the learner. 2 types of self-explanation that I'll focus on in my talk today. One is open-ended self-explanation, and the other is focused self-explanation. So open-ended self-explanation, it provides no assistance to learners when creating their explanations. Learners are expected to create explanations based off of very broad prompts. For example, explain what you learned about the text. The problem with this is sometimes students struggle with selecting that information, which could make creating an explanation more difficult. how to solve that problem is by using something like focused self-explanation. This is a type of self-explanation that provides assistance by indicating to learners what information they should be focusing on in their explanation. For example, explain what you learned about reflected energy in the text. There's been a lot of prior research looking at self-explanation. There's been research looking at self-explanation versus control conditions, different types of self-explanation compared to one another, and self-explanation versus other kinds of strategies. And all of this research has shown that self-explanation is very beneficial to learners, and more assistance tends to be more beneficial to students. However, what's missing from research is really getting at this generative component. Is the fact that students are self-generating it important, or is just having a second explanation beneficial to students? Additionally, a lot of the prior research has been focusing on immediate tests. So there's not much research looking at delayed tests, which would be more beneficial to knowing how self-explanation could help in a more normal learning situation like a classroom. So this, the fact that this is missing from the literature is what inspired this research. So in my first experiment, I designed a 4x2 between subjects design. My first independent was explanation type, which I'll in a few slides. And my second independent variable was time of post-test, where I had an immediate post-test and a delayed post-test. My procedure started with a pre-assessment where I gathered information about the learners, including their prior knowledge in greenhouse gases. Next, I had a lesson where I gave the students a lesson about how infrared radiation is created, the reaction between greenhouse gases and infrared radiation, and how more greenhouse gases causes more heat. And here's a demo of that slide. So as you can see, there's some animation, and at the top of the screen there is text. And students saw 4 of these slides with different information on them. And then between each slide, there were different prompts. So this is where the explanation type independent variable came in. So the first condition, the open-ended self-explanation group, saw prompts such as, please explain what you learned in the last clip. The focused self-explanation group saw something like, please explain what you learned about absorbed energy and reflected energy. The given explanation group saw something like, energy that comes through the atmosphere from the sun can either be reflected off the surface of the earth as reflected energy, or absorbed in the surface of the earth as absorbed energy. And lastly, we had a control where there was no explanation. They were just asked to move forward in the lesson. After the lesson, half of the students got an immediate post-test where they took the post-test right after the lesson, and the second half got a delayed post-test where they left the lab and came back a week later to do that post-test. And the post-test included 7 open-ended questions to assess understanding. And lastly, they did a post-questionnaire that assessed their subjective experience with the lesson. The predictions from my first study were that self-explanations would do better than the control group. The given explanation group would not do different from the control group. The self-explanations would do better than the given explanation group. The focused self-explanation group would do better than the open-ended self-explanation group, and the results would be stronger in delayed test. So when I ran my study, I got my results, and I found that there was a main effect of explanation type, with the open-ended group doing significantly better than the given and the control group, and the focus group doing significantly better than the given group. Additionally, I found a main effect of time of post-test, with the immediate group doing better than the delayed test group, and I found no interaction. However, this didn't really get at the question of, are there differences in the immediate test and the delayed test? So what we did is we ran 2 separate ANOVAs, and when we looked at the immediate test, we found that there was no main effect of explanation prompt. All the groups did similarly. However, when we went to the delayed test, we found the results that the open-ended was better than the given and the control, and our focus group was better than our given. So back to my predictions. We did show that self-explanation led to better learning than the control. The given and the control were not significantly different from one another. The self-explanations did better than the given explanation group. The focus did not actually do better than the open-ended, or the focus group did not do better than the open-ended self-explanation groups, and the results were stronger in the delayed test. In fact, they were only shown in the delayed test. So this led us to the second experiment where we thought, we still think that students struggle with creating explanations. So how do we make a given explanation more generative and more beneficial to students? So what we did is we took the same study and kind of reworked it a little bit. So we had 3 of the same conditions for our explanation type, and we replaced one of them with a new condition called rewrite explanation, which I'll explain in a few slides. The procedure was exactly the same. However, this time we only had a delayed post-test. We did not do an immediate post-test. Like I said, the prompts for the open-ended self-explanation, the given and the control were all the same as experiment 1. However, our rewrite group was new. We gave them the same explanation that we gave in the given condition. However, we added this sentence at the bottom that said, please rewrite this explanation in your own words on the sheet provided. And this added a generative component, hopefully, to this lesson. The predictions for this study were that the self-explanation and the rewrite group would do better than the control. The given group would not do differently from the control. The self-explanation and rewrite group would do better than the control. And the self-explanation group would do better than the rewrite group. When we ran our study, we found that there was a main effect of explanation type. Our self-explanation group, once again, did significantly better than our given and our control groups, and our rewrite group did better than our control group. So back to our predictions, the self-explanation and the rewrite groups did do better than the control. The given did not do significantly different from the control. The self-explanation did do better than the given group, however, the rewrite group did not do better than the given. And lastly, the self-explanation group did not do better than the rewrite group. So what is this telling us altogether? Basically, it's telling us that self-explanation is better than a given explanation. This is showing us that the generative component of self-explanation is what is beneficial to learning. It's not just having a second explanation that is helping learners. Additionally, it shows us that given explanations don't really help learners. In fact, when we gave explanations, students didn't seem to do much better than students that were just shown the lesson itself. Additionally, this shows that self-explanation does benefit students in the delayed test, and this shows that self-explanations have robust effects, which may indicate that generating explanations may help with students' understanding of material. However, for the study, some limitations is that it was a pretty easy lesson, so that might have been why we didn't see any differences between certain conditions. It was also a short lesson, so we're not really sure how this would translate over to a longer course, like a semester or a quarter. And time of post-test was also a potential confound, where those in the generative conditions did take significantly longer than those in the non-generative condition. Although to address that, we would have had to create a lesson that was not super ecologically valid, so we didn't want to address that at this point in the study. For future directions, I think this would be interesting to look at with more difficult lessons to see maybe there'll be more differences between the groups if the material is more difficult to grasp. Additionally, I'd like to look at how the mental models of learners are manipulated using self-explanation. There's theories that state that the mental models of learners, when they're using self-explanation, can either fill in gaps that the students have, or it can help correct any misconceptions the students have. And lastly, I'd like to look at this with learners with less experience in explaining. I think that rewriting could definitely help learners that may not be able to explain very well because they just don't have the experience in it. And lastly, I'd like to give a special thanks to my advisor, Richard Mayer, for helping me with this study and getting it all done. I'd like to thank the Mayer Lab and the Haggerty Lab for giving me great feedback always, and my lovely RAs for helping me collect lots and lots of data. And here are my key references. Thank you.
Thank you, Elisa. Can you hear me okay?
Yes, I can.
Okay, great. We've got some questions. So the first one is from Mike. Okay, terrific work. First of all, why do you think it takes time, as shown by the delay test, for self-explanation to have an effect?
That's a great question. So this is something that I thought about. And I think that part of the reason is because in an immediate test, everyone can kind of just regurgitate the information. You don't really need to have a deep understanding of the material to really do well on an exam. However, you need to have some sort of understanding to hold on to that information over a delay period. You can't just regurgitate the information over a delay. So those that may not have as much of an understanding may not do as well on a delayed test. So that's why I think a delayed test is really important to look at when you're trying to understand how to learn.
Okay. Yeah, we got the same question from Ashley. So
Oh, okay.
Ashley, sorry, we skip this question. So we got another question from Zoe Lieberman. Did you code people's explanations for accuracy? What would you expect when people create their own explanations, but those aren't actually accurate? Or how do we get students to generate good explanations by themselves?
Yeah, so we did actually code people's explanation accuracy, and we did do some correlations with how well they did on the post test. We got mixed results between the two studies. The first one, we found that writing a better explanation did help on the post test, but in the second experiment that we ran, we didn't find that there was a correlation between good explanations and performance. So we're not really sure why or what may be the benefit of writing a good explanation versus just an average explanation. As for prompting people to create good explanations, I think that is where the continuum comes in play. So having an open-ended self-explanation is going to be much more difficult for students to create good explanations. So adding a little bit more assistance is probably going to be more beneficial. And that's what prior literature shows, is that having more assistance in creating learning, especially with those with lower prior knowledge, can be more beneficial. But we didn't really assess that because I think that our material is too easy for students to. It was too easy to learn.
Okay, we got two more very good questions. Casey, let me know if we are running out of our time. So another question is from Vanessa Woods. Do you think reflection is an important process related to generative learning?
I mean, I do think that reflection is important. I think that there's a lot of components that are important to generative learning and generative learning strategies. And I think reflection can be seen as kind of like maybe more of like a metacognitive strategy that gets people to know, okay, is there something that I don't know about the material? And how can I maybe fill that in? And different generative learning strategies do try to get at that metacognitive skill to have people engage in more like reflection, I guess, or more self-assessment of what they know.
Oh, okay. I think another question is quite similar to the one before. So just quickly, just a quick question. Did you look at whether there was a relationship between the quality of the explanation and the learning outcomes? If quality does matter, how could instructors guide learners in generating good quality explanations? So I think this question has been asked before, but do you want to add anything?
No, basically we got mixed results about the relationship between quality of explanation and the learning outcomes. So I think that's something that should be assessed in future research, maybe along the lines of the mental models, understanding how maybe different people explain it different ways because they're trying to fill in different gaps in their mental models. So maybe it's hard to assess what is a good explanation for each person or for everyone, because maybe it's very individual. But I'm not sure. I don't have the answer to that at this moment.
Okay, great job. Thank you.
Thank you.
Okay. Now here to present the Richard E. Mayer Award for Outstanding Research in Psychology, please welcome Professor Hee-Jung Kim.
Hi, good morning. Can you hear me? Okay. All right. So every year, this is my pleasure to do this. And I am here to present the Mayer Award. And the Mayer Award is an award that is established in 2013 to offer a recognition for the student who wrote the best second year paper. And this is annual award. And this year, the awardee, the person who's chosen as the most worthy among very worthy, the competition was Paige Harris. And wish I can hear some applaud, but I imagine that I'm doing so. And so basically the committee, the Graduate Affairs Committee, read a few nominated papers, and we decided that Paige's paper was done very, the Paige's paper represent the excellent research and as well as excellent writing. So it's my great pleasure. And in the past, when we do this in person, we usually present a very fancy plaque, and then some hugs and a lot more confetti and stuff like that. But we cannot. Hopefully, I'll see you soon and give you that hug. Okay, congratulations.
Thank you, Hee-jung.
Congratulations, Paige. We will now have our 10-minute coffee break. Hi, everyone. We are now done with our coffee break. Welcome back. I will now pass it over to Rami.
Okay, can people see me? See the slide? I'll take that as a yes. Great. Our next speaker is a graduate student in James Rooney's Human Behavior Endocrinology Lab in the area of developmental psychology. She is from Beijing, China, living close to its Olympic Park. She earned her undergraduate degree from right here at UCSB, and in her free time, she enjoys doing karaoke. Now, if you're fluent in Mandarin, like I am, you would know that the surname Mei is a negative prefix, meaning that anything good that comes after it becomes bad. So this presenter's father thought of the clever idea of making her first name Mei, so that her full name becomes a double negative and thus a positive. True to her name, she has been a positive influence in the lives of those that she has interacted with. Please welcome Mei Mei.
Can everyone hear me? Okay. Okay. Thanks for the introduction, Remy. I'll be talking about the relationship between women's ovarian hormone and their odor attractiveness in today's talk. So, as many of you guys know, our furry friend like to take a good sniff at each other.
Hey, May.
Yes.
I don't know if did you start your slideshow?
Yes.
Okay, it's still showing it from like the PowerPoint, so maybe stop sharing your screen and try again. So all we're seeing is still the regular. We're not showing the show yet.
Okay, let me do that.
Sorry to interrupt you.
Yeah, that's all right. How do I unhide this? Stop. Can you see it now?
Yes, we can. Thank you.
Okay, so as many of you guys know, our furry friend likes to take a good sniff at each other when introduced to a new member of the opposite sex. So what information are they obtaining through odor? Well, researchers have shown that in lots of mammals, females use olfactory cues to communicate to males important information about their reproductive status. Ovarectomy makes the odor of female metal vow unappealing to males, but the scent attractiveness is restored after injection of estradiol. Research with rhesus macaques also found a similar positive effect of estradiol and additional negative effect of progesterone on odor attractiveness. And male stump-tailed macaques show testosterone response to the odor of late follicular female. So naturally you want to ask, what about human? Can men detect ovulation cues in women? And do ovarian hormone in women predict odor attractiveness? In humans, women's odor samples collected during the late follicular phase, aka the fertile window when conception is possible, are on average rated as more attractive than samples collected from the luteal phase, the time after ovulation when conception is not possible. Men also show testosterone increase after smelling women's odor collected near ovulation, although there are some mixed results on this finding. And a recent study by Lotmeyer and colleague have shown that between women, estradiol and progesterone predict odor attractiveness for samples collected near ovulation. So apart from the Lotmeyer paper that tested the between women effect of hormones, none of these previous research tested any of the hormone effects. And none have tested whether women's within cycle shift in hormone predict the within woman shift in odor attractiveness. And this is what we're looking at in the current study. With that goal in mind, here's a graph of estradiol and progesterone throughout the menstrual cycle. Estradiol start low at the beginning of the cycle, rise during the follicular phase, peak in the late follicular phase, then drop just before ovulation and rise to a second peak in the luteal phase. Progesterone, on the other hand, stay low during the follicular phase and rise following ovulation, peaking in the luteal phase. Therefore, it's unclear whether the decrease in odor attractiveness during the luteal phase found in previous study are due to the decrease of estradiol or the increase of progesterone in the luteal phase. And this is something we want to clarify in the current study. The situation becomes more complicated when we take into consideration the fact that these hormones also vary between women or within women between cycle and tend to be elevated in cycles with higher fertility. As you can see in this graph, if woman A experiencing a particularly fertile cycle, she may have higher luteal phase estradiol, point A, than woman B's fertile window estradiol, point B. Therefore, if odor attractiveness track estradiol only, it should be higher in point A than point B, which is inconsistent with the notion that odor provide clear clue of ovulatory timing. However, if progesterone effectively shuts down odor attractiveness, then women's sample from the fertile window should consistently smell better than their samples from the luteal phase, regardless of cycle phase variability in fertility. Therefore, a positive effect of estradiol and a negative effect of progesterone on odor attractiveness would support the idea that odor provides direct clues of women's ovulatory timing. But if odor attractiveness is coupled with estradiol but not progesterone, then it's possible that some women's luteal phase odor may smell better than some other woman's late follicular odor, or the same woman's late follicular odor from a less fertile cycle, which suggests that odor are cues of general reproductive status. To sum it up, there are two questions we're trying to answer in the current study. What's the relationship between estradiol and progesterone and women's odor attractiveness throughout the cycle? And what's the function of this relationship? Is it a signal of within cycle fecundity, meaning are men detecting direct clues of ovulation, or is it for general reproductive status, meaning is this woman having an ovulatory cycle right now and will she have an ovulatory cycle in the near future, as these cycles tend to cluster? Consistent with previous literature, we hypothesized that odor collected during the fertile window will be more attractive. We also hypothesized a positive effect of estradiol, but no effect of progesterone on women's odor attractiveness. This is because pair bonding in human may have led to evolution of preference mechanism that seeks cues of long-term health and fertility over cues of whether a woman can conceive on that exact date. A total of 46 naturally cycling women were recruited from UCSB. They were asked to collect odor samples by wearing an underarm cotton pad overnight every 5 days over the course of 30 days. So we have a total of 6 samples per woman. On the odor collection day, participants provide a saliva sample that were acid for estradiol and progesterone. And every day, they take a luteinizing hormone test, which is a urine test that they can take at home. Positive LH tests indicate ovulation within about 24 hours. And the other sample that falls between 5 days before and 1 day after the positive LH test is considered a fertile window sample. Odor samples were rated across 5 different rating sessions, with each man rating about 54 to 60 samples. Male raters were asked to sniff individual samples from a glass jar and rate the odor for pleasantness, sexiness, and intensity using a 7-point Likert scale. We later combined the pleasantness and sexiness rating to create an attractiveness composite, as these two ratings are highly correlated. For the first part of our results, we're using women as a unit analysis to analyze data from the donor woman's perspective. For each odor sample, we average across the rating of every rater to get a mean rating for the odor sample. Then we constructed a 2-level hierarchical linear model to analyze our data, with odor sample at level 1 and donor woman at level 2. We'll first talk about the fertile window effect on odor attractiveness and then take a closer look at the late fertile window. We'll then talk about the within woman effect of estradiol and progesterone, and then proceed to the between woman effect of these hormones. So, does cycle days predict odor attractiveness? Let's look at this plot of cycle days on the attractiveness composite. We coded the day of ovulation as day 0 and coded cycle days relative to the estimate day of ovulation. Fertile window days are coded -5 to 0, and we found a no effect of fertile window on odor attractiveness. However, as you can see in this plot, there's clearly a spike in the odor attractiveness in the late fertile window, day -2 to 0. So maybe this effect is more specific to just the late fertile window days. They will run the same test again, but this time with late fertile window days. As expected, when we tested for the late fertile window days only, there was a significant effect on odor attractiveness. This suggests the effect might be more specific to the late fertile window rather than fertile window in general. Next, we want to know what explains the late fertile window effect. Does within cycle changes in estradiol and progesterone predict within cycle changes in hormones, odor attractiveness? Looking at the results, we didn't find any significant effect of estradiol, progesterone, or their interaction on odor attractiveness. Therefore, we didn't find any significant within cycle shift in hormone that might explain this late fertile window effect. Next, we want to know if estradiol and progesterone has a between woman effect. So, we input mean estradiol of each woman at level 2 to test the relationship with mean odor attractiveness. And we found that women with higher mean estradiol have higher mean odor attractiveness across her sample. We then did the same thing with mean progesterone and found a no effect. Together, this no effect of progesterone and the positive effect of mean estradiol provide support to the idea that female body odor provide cues of general reproductive status. For the second part of results, we'll be analyzing the data from the male rater's perspective, examining how detectable late fertile window signal is to men. Having a significant late fertile window effect does not mean that the signal is detectable. So this graph shows the rating of all the samples of each woman donor with donor ID on the x-axis and attractiveness rating on the y-axis. Red dots indicate late fertile window samples and black dots indicate samples from the rest of the cycle. For any approach threshold we set, there will be hits and false positive. And if a man is using odor to determine whether a woman is in the late fertile window, and he set his threshold at the blue dotted line, then all dots above the line indicate a woman attractive enough for him to approach, or in other words, currently in the late fertile window, according to his judgment. Then all the red dots above the line indicate hits, and all the black dots above the line indicate false positives. And notice that false positives seems to outnumber hits, although red dots are on average higher than the black dots on the graph. An ROC curve plot the relationship between hits and false positive across various decision thresholds. In this graph, the diagonal line represents chance performance, which is interpreted as no discrimination ability on the basis of rating. And the top curve shows moderate detectability based on hits to false positive ratio. Here's the ROC curve of the late fertile window effect. As you can see, the late fertile window detectability is very low, not significantly different from chance performance, despite that there was a significant effect in our HLM model from earlier. To conclude our results, although we didn't find any effect of fertile window, we found a significant late fertile window effect. However, this information might not be accessible for men meeting the woman for the first time, as shown in the ROC curve. Additionally, this within cycle variability cannot be explained by estradiol or progesterone, as we didn't find any within cycle effect of these two hormones. We did find a between women effect of estradiol on odor attractiveness, which seemed contradictory to the lack of effect within women. This might be because there are just not enough changes within women to detect this effect. Also, it's also possible that estradiol have a longer term effect, but not an immediate effect on odor. And since within cycle shifts can be explained by the two hormones, there might be something else that's driving this effect. A limitation of the study, we only collected samples for a 30-day period, catching 1 menstrual cycle per woman. So we didn't measure the within woman between cycle variability. Also, Future research is needed to explain what's driving the increase in odor attractiveness during the late fertile window. Maybe it's some other hormone like LH or oxytocin. Finally, for the between women estradiol effect, it's possible that healthier women both smell better and produce more estradiol, which is relevant to men's long-term mate choice. Or maybe the long-term exposure to estradiol increase odor attractiveness, even if the day-to-day variability have smaller effect. In conclusion, the between women effect of estradiol replicate Lohmeyer and colleagues' work. This suggests that odor attractiveness might be signaling general reproductive status. However, the late fertile window effect suggests that there might also be some information about within cycle fertility. But there is currently no evidence at all regarding which hormones regulating this effect. Finally, thank you to all my collaborators and all the RAs in my team.
Congratulations on such a great talk, May. We have a question here for you. Have you looked at any characteristics of the male raters, e.g. relationship status, orientation, age, etc., that might influence their rating?
Yeah, we did look at, it's not in this analysis, in the regression model that we report because it's from the woman's perspective. We also run different regression model from the men's perspective, and we did look at men's SOI score, and it didn't seem to have an effect on their rating on odor attractiveness.
And then from Vanessa Woods, which hormones are you interested in looking at next?
I think maybe oxytocin, because oxytocin also do peak in the fertile window, and it'll be interesting to look at that. The only thing is oxytocin does require a lot of saliva to assay, so it could be hard to run this study.
And then Mike Miller asks, if you were to measure attractiveness and all available cues, including visual appeal, do you think odor would even register as a significant cue?
Yeah, that's a great question. So in the actual rating, the men are rating, so it's a 1 to 7 Likert scale. Most men don't like this odor. So it's really like bad and less bad. And this could just be because human body odor are just nasty, and a woman who's ovulating might smell slightly better, but not like flower, not super good. So yeah, odor cue probably doesn't play a role in men's impression on how attractive this woman is. It's more likely that face and body might play a larger role, but it is part of the whole package of the attractiveness fluctuation throughout the cycle.
Is it possible that these effects are reduced in current society compared to our evolutionary history because of the use of cosmetics, scented soaps, etc.?
Yeah, so in our study, we asked the participants to shower with the unscented soap we provide them with and not wear any deodorant on the day of odor collection. But yeah, in the current modern society, men may, at least the smell of cologne and perfume might overwhelm the smell of body odor if the woman is wearing any perfume.
We have one more question, I believe, as of now. Nancy Collins asks, in the mating world, would odor interact with women's behavior cues of potential of interest in the man?
Women's odor interact. Can you repeat the question?
No problem. In the mating world, would odor interact with women's behavior cues of potential of interest in the man?
Yeah, I mean, if the women are actually more attractive, say she's more attracted in the fertile window, she could also be, more flirtatious and more interested in men during the fertile window. Those two probably would, I would predict that those two would change in a similar direction.
One more question just came in from Madeline Gross. Could it be that the effects of odor on attractiveness are happening at the subconscious level in the men?
Yeah, it's possible. Actually, we have a follow-up study with the same data, with the same odor sample we collected. And that study measured men's testosterone and cortisol response after smelling the odor. So that would hopefully capture some of the unconscious level of women's attractiveness changes.
Okay, I don't think we have any more questions as of now. There is still some time left if anyone would like to ask any more questions.
Okay, we can hand it back to the MCs then and move on to the next presenter. Thank you, May.
Great job, May.
Thanks.
Thank you, May. So our next, oh, sorry, let me share the screen. Our next presenter is a graduate student in Hee-Jung Kim's Cultural Psychology Lab in the Social Psychology area. She is from Menlo Park, a few hours north of Santa Barbara, and is also an alumna of UCSB. In addition to her athletic accomplishment of running a marathon in her first quarter of graduate school, she has a fondness for animals, having fostered over 20 kittens and 6 adult cats. As you can see, her passion for food is rivaled only by her passion for research, as evinced by her being awarded the NSF's Graduate Research Fellowship. Please welcome Vicky Papadakis.
Screen. Can you guys hear me okay? All right. Hi, everyone. Today, I'm going to talk to you about some research I've been doing on social class, social tuning, and pro-environmental behavior. So our modern world is faced with an environmental crisis. Both the diversity of our planet and our own livelihood are threatened by this crisis. Scientific consensus points to humans as the cause of this, so rectification of the problem also lies in changing human behavior. People do have different opinions about climate change, though. If you saw this person walking down State Street, you might not pay too much mind. This messaging on his t-shirt seems to be pretty consistent with the narrative we hear about the environmental crisis in our area. However, if you saw a person wearing this t-shirt walking down State Street, how might you react? This is different environmental messaging than we might typically see while walking downtown. How you react to this social cue might also depend on your culture, specifically the culture brought about by your social class. Social class refers to hierarchical social categories of which people occupy different spaces. These different class categories afford people different cultural value and meaning systems. This has implications for people's worldview and their self-controls. Specifically, those from higher social class backgrounds tend to be more independent, whereas those from lower social class backgrounds tend to be more interdependent. Independence-interdependence is a dimension of self-control that affects self-other connectedness. Those from more independent backgrounds tend to have conceptualizations of the self that are bounded with little to no overlap with others, meaning that the self is more separate from others. Those from more interdependent backgrounds tend to have conceptualizations of the self that overlap with close others, meaning that The sense of self includes and incorporates others. Research has found that self-control has a variety of behavioral effects. One of these effects is conformity, or the way that people adjust their behavior to group-level norms. Research has found that those from more interdependent backgrounds tend to conform more than those from independent backgrounds. While conformity occurs with group-level norms, social tuning refers to an individual's adjustment to another individual's beliefs. As such, social tuning occurs at more of an individual level and refers to people tailoring their behavior to fit with an interaction partner in a more interpersonal context. No previous research has looked at how people from different cultural backgrounds engage in social tuning. So how might social tuning be affected by social class and self-control, specifically in the context of pro-environmental behavior? There's been some research relating social class, conformity to norms, and pro-environmental behavior. Some research that looked at donations towards a pro-environmental cause found that for those from higher social class backgrounds, their personal environmental beliefs most strongly predicted their pro-environmental donations. For those from lower social class backgrounds, however, the perceived norms of their communities most strongly predicted their pro-environmental behaviors. With this research on conformity, social class, and pro-environmental behavior, how might social tuning, which is similar to conformity, but at more of an individual and interpersonal level, be implicated? So the present research plans to answer this question. The proposed theoretical model is that experimental condition will lead to different types of social tuning, and that this will be moderated by social class. Social class, in turn, will lead to different levels of self-control. So in other words, the moderation by social class will be mediated by self-control. Our first hypothesis is that those from lower social class backgrounds will be more likely to align with social cues regarding pro-environmental action. Specifically, this means that those from lower social class backgrounds will engage in more pro-environmental behavior in the pro-environmental condition, less pro-environmental behavior in the anti-environmental condition. And we don't expect to see such differences for those from higher social class backgrounds. Hypothesis 2 is that the effects of the first hypothesis will be mediated by self-control. So in other words, the moderation by social class will be mediated by self-control. Specifically, we're interested in the self-control dimension of independence and interdependence. Our participants came from the UCSB subject pool, and the demographics were quite typical for this sample, as you can see here. In terms of how we operationalize social class, we use first versus continuing generation college student status. So given that our participants were current college students, we looked at their family background, specifically whether or not their parents attended a four-year institution. If at least one parent attended a four-year institution, the student was considered continuing generation, and if neither parent attended a four-year institution for college, the student was considered first generation. This is a typically used method for operationalizing social class, especially in the context of college students. In the present study, we followed the basic paradigm used in social tuning research. Participants were greeted by a research assistant wearing a different t-shirt. They were then led into a room and spent the remainder of the study on their own on the computer. Participants completed several questionnaires and then were told that the study was over, but that there was an opportunity to play a game to raise funds for pro-environmental organizations. If participants decided not to participate, they were debriefed at that point. If they decided to participate, they engaged in the activity and then were debriefed. So the different conditions in the experimental manipulation were pro-environmental, where the t-shirt read reduce, reuse, recycle, anti-environmental, where the t-shirt read that global warming was a hoax, and a control t-shirt where there is no messaging at all. During the survey portion of the experiment, we measured participants' environmental belief, independence, and interdependence. One thing that's important to note here is that we treat independence and interdependence orthogonally. In all analyses where we look at independence and interdependence, we look at them as orthogonal constructs. Additionally, we measured people's self-report pro-environmental tendencies, but we won't focus on that for this presentation, and we'll instead focus on a behavioral measure of Boggle, which I'll talk about now. So at the end of the study, participants saw this message on the computer. The message basically asked participants to volunteer to play a simple word game to raise funds for pro-environmental organizations. And participants were able to say yes or no to this volunteer opportunity. If participants said that they were interested in volunteering, they were presented with this word game. They were told that for each word found, donors would contribute 5 cents to pro-environmental organizations. This boggle game served as a behavioral measure for pro-environmental action. We looked at both whether people agreed to participate in boggle as a dichotomous measure of our DB, and we looked at it by number of words generated as a continuous variable. So we first ran a 2x3 ANOVA to see if social class and condition interacted to affect pro-environmental behavior in this boggle activity. So this interaction was not significant, which means that there was no effect of the t-shirt the experimenter was wearing and participants' social class on the number of words found in boggle. So further analyses showed that social class did not predict self-control in the way that we were expecting, based on the literature. So both first generation and continuing generation students had a similar pattern of results and were more independent than interdependent. Because of this, we went straight to the moderator, self-control, and examined its effects. So we first looked at whether or not people volunteered to play boggle as a dichotomous version of our DV, but nothing quite came from that. But when we looked at it as continuous, these are the results depicted here. So we looked at the independence dimension of self-control. What you can see in this graph is that there was movement in the anti-environmental condition for highly independent people, such that they donated more in the anti-environmental condition. In other words, the anti-environmental experimental condition resulted in increased pro-environmental behavior for those who are high in independence. We also looked at interdependence, but that didn't quite moderate either. Okay, so to summarize, we expected that those from lower social class backgrounds would socially tune more than those from higher social class backgrounds. However, we didn't quite find that. What we did find, though, is that people with more independent self-controls were more willing to go against norms in the social environment based on the t-shirt the experimenter was wearing, whereas those with less independent self-controls did not show this pattern. A major limitation to this research is the sample. So we had UCSB college students as our sample, and on UCSB's campus, environmental belief is generally very high. So future research should look at these effects with a community sample where there might be more variability in environmental belief. And in better elucidating these effects, we may be able to leverage these findings to increase pro-environmental behavior. Thank you very much.
Nice job, Vicky. So we have a question from Mary Haggerty. Do you worry that the participants guessed what the experiment was about?
So we asked at the end of the experiment to see if people caught on to anything, and most didn't actually even notice the t-shirt. But social tuning is something that is supposed to kind of happen subconsciously anyway. So that wasn't something that we were super concerned about. But yeah, most people didn't realize what the experiment was about, surprisingly.
Nancy asks, did you also look at cultural differences in ethnicity?
We have that data, but we haven't looked at that quite yet. But yeah, that will be interesting to look at.
All right, thank you so much, Paige. Or sorry, Vicky.
No worries. Thanks.
Great job, Vicky. I would now like to introduce Dr. Michael Gord, who will present the Carlisle Award.
Hello, can everyone see and hear me?
We can.
Great. So hello, everyone. It's my great pleasure to present the Harry J. Carlisle Memorial Award to graduate student Kevin Sitt. So the Carlisle Award, for those who aren't familiar with it, it was established in 2003 in recognition of the important contributions made by Professor Carlisle, a longtime faculty member in our department who retired in 2001. So each year, this award is presented to a graduate student in the NB area who exemplifies the qualities valued by Professor Carlisle. So namely, sound scholarship and research, as well as good citizenship and concern for their fellow students. I personally can think of no one kind of more deserving for this award than Kevin, who's really been a star in both the classroom and the lab. So Kevin actually joined my lab when my lab was really just a single room in the basement full of cardboard boxes, and he was really instrumental in getting things up and running. Over the past few years, Kevin has been carrying out some really beautiful research to understand how mice process kind of coherent visual motion and how it influences their internal sense of direction as they navigate through the environment. This research has involved everything from learning neurosurgery to engineering custom microscopes and hardware, develop new computational analyses, which Kevin has really dived into with great enthusiasm. He recently had a very nice paper accepted in Nature Communications, so congrats to him for that. And he has several more exciting manuscripts on the way. So finally, anyone who's interacted with Kevin knows he's really an amazing kind of department citizen as well. I remember one incident in my lab that kind of exemplified the kind of graduate student that Kevin is. There was a very scary occurrence that caused great panic where our long-suffering coffee pod machine suddenly malfunctioned. Now, most graduate students, I think, would have just begged me to buy a new one for the lab immediately. But Kevin instead opened up the coffee machine, pulled out the circuit board, and searched around until he found a burnt out capacitor and soldered in a new one, making sure that everyone was functioning at 100%. So more seriously, though, anytime deadlines were looming and people were stressed, he was always good-natured and happy to help other students with their research or their presentation or their code, or just to joke around for a bit. So I'm very happy to present this award, and I'll wrap it up before I embarrass him any further. But please join me in congratulating Kevin Sid.
Congratulations, Kevin. We will now have our second 10-minute coffee break. Hi, everybody. Welcome back. Our next speaker is a UCSB Crossroads Fellow from Emily Jacobs' lab in CPCN. Before this graduate program, she was actually Dr. Jacobs' very first lab member, where she worked as a lab technician for 2 years. She is an active member of the community where she mentors first-generation undergraduates, and she supports notions of diversity and inclusion on campus through her involvement in the Asian Pacific Islander Graduate Student Alliance Group. Here to talk to us about her research involving sex differences and age-related changes in spatial navigation, Xuying Yu.
Hey, hi everyone. Okay. So I'm gonna assume y'all can see my slides and can hear me. All right, so for my presentation, I'll be talking about some of the behavioral findings from my project on spatial navigation and aging. So to give you some context, oh, is there someone talking? Oh, okay, no, that's fine. Sorry, so to start off, just to give you some context, spatial navigation has emerged. Spatial navigation is a complex behavior, and it encompasses many different aspects. That includes the ability to update your position in space, learn the layouts of new environments, and also plan routes to goal locations. There are reports of many, large individual and sex differences. However, many of these chronological aging studies often fail to address the critical changes that occur before the age of 65. For example, key neural circuitries that support spatial navigation, such as the prefrontal cortex and the hippocampus, are really sensitive to the neuroendocrine changes that occur during the midlife period, especially when women go through menopause. So this is the gap in the literature that we want to address, because it's still unknown whether these neuroendocrine changes can affect brain behavior relationships and supporting navigation behavior. Another motivating factor is that spatial navigation has emerged as a promising detector for dementia. The prevalence of Alzheimer's is 2 times greater in women than in men, which suggests that sex plays a specific role in understanding disease risk. Therefore, taking into account sex differences is important for understanding trajectories of healthy aging. So for our study, we recruited healthy young and midlife men and women to investigate whether navigation differences emerge earlier in the aging process, and also to investigate whether sex differences persist with age. So here's an outline of the 3 main navigation abilities that we examined, and this is to give us a comprehensive understanding of how navigation differs by age and sex, and also to identify which aspects of navigation are more sensitive to aging. So throughout the talk, I'll go through each one at a time, and I'll explain what it is, how we tested it, and what we found. So the first aspect of navigation ability that we tested was path integration. This is an ability to return to a home location after traveling for some distance. So this relies on self-motion information from body-based cues and optic flow. It's been reported that older adults tend to make more errors than younger adults, but we don't know if these age-related changes emerge by midlife or if they differ by sex. So for our study, we test participants by having them put on a headset, and they're in this immersive virtual reality environment, and we have them physically walk in a loop closure task. So participants start by standing behind an orange pole, which is their starting location, and the red arrows indicate the direction that they'll be walking in. So the orange pole will disappear once they walk through it, and that will begin their trial. And the goal is to make one complete loop around the circle until they believe they've ended up back to where that orange pole used to be. And participants click on a wireless mouse to stop, and then they will move to the next trial. And so all of these trials are randomized at different starting positions and at different radius sizes, and trials also alternated between walking clockwise and counterclockwise. Our 2 main dependent measures were position error and degrees traveled. So position error is the straight line distance between where they thought was the start and the actual start of the trial. and degrees traveled reflect how much they walked on the circle. So ideally it would be 360 degrees, but this can, but participants can either be either below or under that number. So for position error, we did not find any age or sex differences, but we did find an effect of radius size. So we can see that participants tended to make more errors as indicated by the increasing distance in meters on the y-axis as the circle size increased, as shown on the x-axis here, which is the radius size ranging from 1 to 3 meters. And for degrees traveled, we also did not find any age differences, but we did find that women tended to undershoot, whereas men tended to undershoot. So the dashed line here at 360 degrees is the ideal degrees traveled. And we can see that women tended to be above that line, whereas men tended to be below it. And similar to position error, we also found an effect of radius size such that as the circle size increased, so did the degrees traveled, such that participants tended to walk more along the circle. The next aspect of spatial navigation ability that we examined was the acquisition of spatial knowledge. So this is also known as wayfinding success, and this is the ability to acquire and configure spatial information to connect known paths to new routes. In the literature, there are reports of a male advantage, and there are also reports of older adults doing worse than younger adults for wayfinding success. However, we want to examine this earlier in the aging process and to examine whether we see sex differences between midlife participants. We measure the acquisition of spatial knowledge using proportion of correct trials on a desktop maze learning task. So here in the overhead view, you can see this is a map of the maze. And during the learning phase, we ask participants to freely explore it and to try to find and remember the locations of 9 different objects. And these are the red dots in the maze. And during the test phase, we ask participants to navigate from one object to another. And so all the objects are obscured in the test phase. So participants will have to rely on their acquired spatial knowledge during the learning phase in order to correctly navigate to the correct target objects. So trials were labeled as correct if they went to the correct target object, and trials were labeled incorrect if they went to the wrong one. So on the x-axis is the age groups depicted by sex, and on the y-axis is our measure of wayfinding success, which is the proportion of correct trials ranging from 0 to 1, and the dashed line at 0.11 represents chance level performance. So after controlling for the number of moves that participants made in the learning phase, we found that midlife adults were less successful than younger adults, and overall, women were less successful than men. We found a significant sex difference between young participants, such that young men made more correct trials than young women. However, we didn't see this sex difference between midlife participants as they performed similarly. Lastly, we tested navigational strategy. So there are 2 main established behaviors for navigation strategy. One is the use of flexible place-based strategies, or known as taking shortcuts. And the other is a rigid response-based strategy known as taking learned routes. And in the literature, older adults tend to rely more on these learned routes than younger adults. And there's also a well-established sex difference, with males preferring to take shortcuts overwhelmingly more than females do. So we tested navigational strategy on a desktop maze to investigate whether midlife adults and women take fewer shortcuts than younger adults and men. So if you look at the overhead view, during the learning phase, we have participants start here at the red dot, and we ask them to follow a learned route, which is this purple line. So they do this route 5 times, and each time they're able to see the different objects, which are the black diamonds. During the test phase, we ask them again to navigate from one object to another in a given amount of time. And participants' strategies can vary. So they can vary from taking shortcuts where they're walking on paths that they haven't traveled before to taking routes that they were familiar with. And to measure their preference for shortcuts, we used a formula called solution index. And this is the number of shortcuts that they took divided by the number of successful trials. So again, on the x-axis, we have the age groups depicted by sex, and on the y-axis is the solution index ranging from 0 to 1. We found that overall, midlife adults took fewer shortcuts than younger adults did, and that women took marginally less shortcuts than men. We also found a significant sex difference between young adults, where young men were more likely to take shortcuts than young women. However, we didn't see this sex difference between our midlife group. And we also generated heat maps for this task just to qualitatively assess the pathways that people took during the test phase. And just as a reminder, here's the overhead view of the map again, showing you that the purple line is the route that they followed during the learning phase. So between young participants, we can see that men were more likely to go into the center of the maze, whereas women tended to rely more on the learned path. Between midlife participants, we can see that both midlife men and women behaved similarly, since they were more likely to rely on the learned route. So in summary, the 3 different tasks and the 3 different navigation abilities provided us a comprehensive understanding of how these different navigation abilities differ by age and sex. In summary, we can see that for path integration, there were negligible effects of age and sex. And even though we found a sex difference for degrees traveled, this effect was similar across young and midlife participants, and it didn't affect the overall position error. For the acquisition of spatial knowledge, we were able to find that these age-related changes could emerge as early as midlife, but then the sex differences that we observed between young participants didn't persist with age. And it could be that because the midlife participants were performing around chance level, that this reflects a floor effect, and it made it harder to detect any kind of sex difference at all. And lastly, for navigation strategy, even though we gave equal exposure to the route for all participants, we found that midlife men and women showed increased reliance on taking habitual paths. And to kind of follow up on these behavioral findings, we hope to eventually implement all of these tasks into fully immersive virtual reality experiments, just to eliminate any kind of computer inefficiency across participants. And we also want to assess performance as a function of reproductive age. Otherwise, or in other words, we also want to see whether pre, peri, and postmenopausal women who might be at the same chronological age differ in their performance level. We also collected neuroimaging data from these young and midlife participants, so we can also assess hippocampal subfield volume, as well as white matter connectivity structures in relation to performance. To conclude, our findings suggest that the acquisition of spatial knowledge as well as strategy use are more sensitive to the earliest stages of the aging process. And by understanding, trajectories of healthy aging and how they differ by men and women, our findings can actually help pave the way for developing behavioral and neural markers for dementia. And that is the end of my talk. I just want to say thank you to my advisor, Emily Jacobs, and my wonderful RAs, as well as members of the Hegarty and the Crastel Lab for their support in this project. And also shout out to Dr. Dan Conroy Beam and Jack for their assistance with R.
Great job, Xuying. Diego is going to take questions for you, and while he does that, I would like to share your baby picture because it is simply too cute to not share. So while he takes questions, hopefully everyone can see that now. Diego, I'll pass it on to you now.
We cannot hear you, Diego. Do you want me to do the questions for you? I'm more than happy to. So I will ask questions because Diego might be having some technical difficulties since we can't hear him. Let's see. So we'll start off with one from Michael Miller, which says, do the declines in navigation with age reflect a specific decline in spatial cognition or a more general decline in cognition?
Oh, that's a great question. So the question is, does it reflect only in navigation performance, or is it just like a general like cognitive decline overall? I don't know the answer to that, but what I would try to speculate is that spatial navigation is the kind of first sign of dementia. And so because this type of behavioral performance might be due to some kind of like evolutionary, like, oh, sorry, I got distracted. It might be due to some kind of like evolutionary like pathways in which our brains might be like this kind of like behavior might be the first to go. And I think spatial navigation, because it does kind of include, you know, different cognitive processes. So it includes the ability to take in perceptual systems, the ability to attend to visual information in the environment and so forth. And so I think it does kind of reflect an overall more general sense in terms of like cognitive decline or overall like cognition.
So can you hear me?
Yes.
Okay, perfect. So I got my microphone to work, so sorry about that. The next question is coming from Nicole Hahn. Does the difficulty of the navigation task affect people's tendency to take shortcuts?
So does the difficulty of the task make people more likely to take shortcuts? I would say no, because if it is like too difficult, I would imagine that people would be more likely to take the learned route, just because taking shortcuts, at least like for the tasks that we have the participants do, we have them follow the learned route. And so they weren't given any kind of experience with traveling to other places in the maze. And so that I would imagine would be a little bit more difficult to have this like cognitive map of the maze so that you can do these shortcuts. So if the task was more difficult, I would imagine that people would be more likely to actually use the learned route.
Okay. And this question is from Hongbo. To what extent the age and sex-related differences in navigational strategy can be attributed to their navigation ability and to other non-spatial traits, for example, risk preferences?
Yeah, I think like with spatial navigation, because it is kind of a very broad umbrella encompassing many different like aspects, that there could be other underlying like behaviors that can be teased apart from that. So one of the things that you mentioned was risk-taking behavior. And there are studies looking to see like how these sex differences the sex differences that we see in spatial navigation ability could be from spatial anxiety or from the ability to just take more risk. And, you know, I think there's a lot of different like factors that can go into this. So there could be like geospatial like locations of how people were, where people were reared, and also like cultural differences in, you know, how women and men might take or prefer like different types of strategies in doing these types of behaviors. So I do think it does kind of reflect other underlying like factors that can be teased apart.
And here's a question from Evan Layer. What do you think causes the sex differences in spatial navigation for young adults?
So kind of like going back to that previous question, I guess. So I think like between younger adults, there have been like kind of like studies looking at also like risk-taking behaviors as well as spatial anxiety. But I know that this task was adapted from Alex Boone, who graduated not too long ago from this department. And he found that like it is probably more of like a preference for what people prefer to do. And so if you're given like this task and you tell people to, you know, just like go to the goal location, for women, maybe they're just more likely to just do it just so they can actually ensure that they get to the actual goal, versus like young men who probably might have like an advantage because, well, not to perpetuate like stereotypes, but it might be that they have more like video game experience. So it gives them the ability to actually create a better sense of the cognitive map of the environment so that they're able to take more shortcuts.
That kind of taps into, or actually mainly answers Madeline's question, so I don't know if you'd like to tack on anything else. Okay, yeah, she said she's already responding in chat saying that's already been answered. So she was asking, did you measure comfortability with the VR and or experience using video games? She was thinking if young men are more experienced navigating virtual spaces in video games, maybe that could explain some of the effects. I don't know if you wanted to tack on any more regarding that topic.
Yeah, I can just add to that. So yeah, definitely. And so with the younger participants, they were given like a little survey at the beginning to just assess like who has more video game experience than other people. So that data is there, but it hasn't been analyzed yet. And this is one of the reasons why we kind of eventually in the future want to implement all of these tasks into just like a headset wearing experiment so that there are no questions about people having like more experience with computer controls than other people might be having.
Cool. And another question from Barry Giesbrecht. Do you think they have no representation of potential shortcuts, or is it that they have it, but choose the one that they know to ensure that they will be correct?
So I would say it's more of the latter, that they choose to use the learned routes. So I know Alex Boone, he also did a follow-up study to his study in young adults where he instructed participants to only take the shortest route. And you can see that like young women were actually able to take shortcuts. It's just a matter that they don't prefer to. For the midlife participants, it's kind of funny because like as they're doing these tasks, they kind of like comment and like say things like as they're like doing these tasks. And I also have like follow-up questions with them about what strategies they used and why. And many of them have told me that, oh, it's only because they are more comfortable using the learned route. And even though they know that they can take a shortcut, they just say like, oh, I'm more comfortable doing this because I know for sure that I can get there.
Awesome. Well, that was the last question. Thank you so much.
Great job, Xuying. Our last speaker is from Barry Giesbrecht's lab in CPCN. He may not have been able to ride a bicycle until his senior year of college, but he was awarded the PBS Regents Fellowship. Outside of academia, he enjoys hiking, working out, programming, and playing video games. In his free time, he even learned how to create AI agents that attempt to play video games themselves. Today, he will be speaking about his research involving tracking the contents of spatial working memory. Here is Jordan Garrett.
Oh, I'm free. All righty, here we go. Hope everybody's doing good. Okay, can everybody see my screen? I'm gonna assume yes. Will, can you see the screen, Will? My man, appreciate it. All right, thank you guys for that introduction. So I know we're on quarantine and the campus is pretty dead right now, but it's not hard to imagine thousands of undergraduates all speeding along the bike path towards their destination. And I'm confident at least a few of you here could share stories in which you were hit or almost hit when attempting to cross these bike paths. And you might have been thinking to yourself, and this is probably like the more PG version of it, what in the hell was going on in that person's brain before they ran me over with a damn road bike? Well, much of what we know about brain function during physical activities such as cycling stems from animal research. For instance, when looking at neurons in layers 2 and 3 of mouse primary visual cortex, it has been shown that there's an increase in neural activity or tuning for preferred stimuli during locomotion as when compared to rest. And this same effect is also evident at the population level response. Now, extending these findings across species, human EEG studies have shown an increase in the early visual evoked response during low intensity cycling. Further, it has also been shown that this same exercise modality elicits a multiplicative gain in the reconstructed orientation selective response. So there's ample evidence for an enhancement of early visual processing when we are in a physically active state. Now you might be thinking to yourself, great, so you told me that they could see me better. That still doesn't explain why they ran me over. Well, although we are seeing these enhancements in sensory processes, little is known regarding how higher order cognitive functions are also modulated during physical activity/exercise. One such cognitive function is working memory. As many of you know, this is our ability to maintain and manipulate information over short durations and serves as a platform for guiding goal-directed behavior. Now, when looking at working memory performance during exercise, the literature is pretty mixed, like myself. For instance, it has been shown that performance is impaired during moderate to high intensity exercise. On the flip side, it has also been shown that performance is enhanced during moderate intensity cycling. And to make things even more complicated, it has also been shown that this same exercise modality has no impact on working memory. So it's pretty unclear on how working memory is being modulated when we are in a physically active state. Plus, prior research largely focused on behavioral measures of performance, yet little is known regarding if the neurocorrelates underlying working memory representations are also modulated during exercise. Now, considering prior work showing the enhancements in early visual processing, we decided to investigate if visual spatial working memory representations are modulated during low intensity cycling. To test this, we recruited 34 college days participants from the university and had them complete a simple spatial change detection task, which consisted of first the presentation of a sample stimulus at one of 8 peripheral locations, followed by an empty retention period, and the presentation of an identical probe stimulus at either the same or a different location. Subjects made responses via mouse press while seated on a stationary bike. Now, using a counterbalanced repeated measures design, subjects completed this task under both a resting and an exercise condition. In the exercise condition, they engaged in low intensity cycling with a resistance of 50 watts and at a cadence of 50 RPM. We ensured that subjects maintain this cadence by having them pedal to the beat of a metronome. So to control for dual task differences that might exist between conditions, we had subjects tap their feet at a similar cadence in the resting condition. Now, to record neural activity, we used a 64-channel EEG cap and ensured that subjects maintained fixation throughout the entirety of the trial period by using an eye tracker. We collected physiological data using heart rate monitor and subjective ratings of perceived exertion by using the Borg scale. Now, a large corpus of work has shown that activity in the alpha frequency band plays a major role in visual spatial attention and working memory. So considering this, we decided to assess the contribution of this frequency band to visual spatial working memory representations during exercise by employing an inverted encoding model approach. And for our statistical inferences, we employed Bayesian paired sample t-tests with a threshold of a Bayes factor of at least 3, which correspond to there being moderate evidence for a statistical difference. First, looking at the physiological data, we observed an increase in heart rate and ratings of perceived exertion in the exercise condition, indicating that we were successful in inducing a physically active state. Looking at the behavioral results, we observed no differences in D prime or response bias between the 2 conditions. Keep in mind, though, that this task is relatively simple and that subjects were performing very well on it in both conditions. Now, moving on to the EEG data, prior work has shown that topographical patterns of alpha power tracks the current stimulus location that is being maintained in working memory. And this pattern is characterized by an increase in alpha power over posterior electrodes ipsilateral to the presented stimulus location. Overall, we observed that patterns of alpha power tracked the presented stimulus location in both conditions. Now, to quantitatively assess the amount of to quantify the spatial information the spatial selectivity, to quantify the spatial selectivity in these patterns, we employed an inverted encoding model. Some of you may already be familiar with this technique, but for those of you who are not, you first begin by generating a hypothetical channel response function for each of the 8 possible locations that a stimulus could appear in. This is collectively known as your basis set. Then, using the GLM, we modeled patterns of alpha power as the best weighted linear combination of hypothetical channel responses from this basis set. During a training phase, we used a portion of our data and derived these weights by using least squares estimation. Then in a testing phase, we took the pseudo inverse of these weights along with patterns of alpha power from left out data in order to reconstruct location-selective responses. These responses were then circularly shifted to a common stimulus center, which we denote here as 0 degrees channel offset, to serve as a reference frame. Note that peak channel response over 0 degrees channel offset indicates that the patterns underlying these responses contain spatial information about the stimulus. If these channel responses were flat instead, then that means that the patterns underlying these channel responses contain little to no information about the stimulus. Now we applied the inverted encoding model at each time point throughout the child period in order to reconstruct a location selective response during both the encoding and the retention period. On the x-axis here, we have time, where 0 represents stimulus onset, 250 represents stimulus offset, and everything afterwards is the retention period. On the y-axis, we have channel offset in degrees, and then on the z-axis, we have channel amplitude. First, looking at the resting condition, we were successful in our reconstruction of a location-selective response throughout the entire trial period post-stimulus onset, which was replicating prior work. More critically, though, when looking at the exercise condition, we were once again successful in reconstructing a location-selective response, although we did observe a decrease in the peak amplitude during the delay period. Now, to quantitatively compare these channel responses between conditions, we first folded them over 0 degrees channel offset and then computed the corresponding slope, which served as our index for the amount of spatial information in the patterns underlying these channel responses. When looking at the slopes, we observed at least moderate evidence for them being statistically different from a permuted baseline in both conditions, as indicated by the yellow and cyan lines here at the bottom. When comparing slopes between the conditions, we observed a decrease in slope in the exercise condition during the delay period, as indicated by the red line here. This suggests that our selectivity for locations being maintained in working memory is decreased when we are concurrently engaged in exercise. Now, it's possible that these effects are just a byproduct of differing levels of alpha power or alpha signal to noise ratio in the two conditions. So we conducted control analyses in order to test the validity of our results. When first looking at alpha power, we observed no difference between the two conditions throughout the entire trial period. Further, when looking at alpha signal to noise ratio, which we define as alpha power divided by the standard deviation of power in neighboring frequencies, we observed no differences in the distribution along with the levels of alpha power between the two conditions. Thus, it's likely that our observed effects are not a byproduct of these potential confounding factors. In summary, it is possible to reconstruct a location-selective response during exercise, and that while we are concurrently engaged in exercise, our selectivity for locations being maintained in working memory becomes degraded. Now, our behavioral results did not show the same ones that we observed from our inverted encoding model results, but keep in mind that the task is relatively simple, and it's possible that ceiling effects are masking any differences. Thus, concurrent exercise may have a detrimental impact on working memory performance. So what may have happened is that wannabe hot rod cyclist that ran you over just had a poor representation of your current location. Or it could have just been that you gave them a bad grade on a midterm and that they were coming at you with a vengeance. I'd like to thank my advisor, Dr. Barry Giesbrecht, for supporting me throughout this entire project. He is phenomenal. I'd also like to thank my project scientist, Dr. Tom Bullock. Honestly, without him, I'd probably still be on my first subject. I'd like to thank my other project scientist, Dr. Mary McLean. She's really missed right now. She's currently working the job somewhere else, and it's pretty fresh and pretty raw. I don't want to talk about it. I'd also like to thank the rest of the Attention Lab, especially my fellow graduate students, Dia, Chris, and Ann, for guiding me throughout these past 2 years and making them just a wonderful blast. I'd also like to thank the renowned research specialists that many of you already know, Jamie, Alex, and Leanne, especially for letting me mooch off of their participant pool that they recruited for another study. So without them, I'd probably still be on like my first 5 subjects. I'd also like to thank my research assistants, Owen, Austin, and Dana, and Cameron, not only for participating in my study, but also for helping me run subjects, especially on those 2-a-day weekends. And with that, does anybody have any questions, comments, and concerns? I messed up the joke at the end there, dang it.
Hi, Jordan. I will be your question handler.
Thank you so much.
Absolutely. Best for last, so let's do it. So Michael Miller starts with, if evolution led to higher visual response during extreme exercising, why would it lead to lower working memory at the same time?
That's an interesting question. I've actually discussed this a little bit with Mei slightly during my first year. I would think that it could just be due to some amount of cognitive demand. I mean, we don't really know what are the causal sort of like what's causing these possible degradation in working memory performance during exercise. It could be that there's a lack of communication due to just so much noise going on in the brain when you're concurrently exercising. It could be that levels of neurotransmitters, because like acetylcholine and other catecholamines increase when you're concurrently engaged in exercise. It could be that they're not at the optimum level, which causes there to be miscommunication between regions. But to be honest, it's just purely speculative on why working memory performance would be degraded when we are seeing these enhancements in visual processing or other sensory processes, which is something that we want to look at a little bit further. But sorry, I don't have a good response for you, Mike. Great question, though.
All right, our next question is from Vanessa. She says, do you think the effect you found is specific to visual working memory, or would you expect to get the same effect in verbal working memory?
No, that's an interesting question, because working memory, as many of you guys know, it's a multimodal sort of like cognitive domain, and so it's possible that each of these different domains like visual, verbal, and auditory working memory are selectively impacted by exercise. But for the most part, it seems as though during concurrent exercise, there's been decrements in executive function in the literature, but the literature is pretty mixed. A lot of people didn't control for possible dual task differences like we did in our study. So I would think that it might be something that is possibly uniform across different domains of working memory. I mean, we only see these enhancements. I haven't looked into other possible sensory processes, but we are seeing these enhancements in visual processing, and yet we are seeing these decrements in working memory. And so if we are seeing enhancements in visual processing, but we aren't seeing enhancements in such like auditory processing or other sensory processing domains, then it's likely that upstream cognitive abilities that are dependent on these sensory processes would also be degraded.
All right, the next one is from Mary. She says you answered part of her question or half of her question, so I'm still going to ask it so you can add to the next part. It says, do you think that exercise would have an effect on more complex spatial working memory tasks and or other aspects of working memory? Sorry, I moved. Of working memory, such as verbal words, WM, words per minute. Is that what that is? WM or verbal WM?
So some of the studies that I showed were sort of like verbal working memory studies, and the literature once again is pretty mixed in terms of like showing enhancements of verbal working memory or decrements in verbal working memory. So I think they definitely modulate performance. It's just really going to be dependent on the exercise type. Like we chose cycling because prior work has shown that this exercise modality is the one that elicits the most enhancement, or most frequently elicits an enhancement in cognitive processes. So I do think that exercise would modulate other domains of working memory. And in terms of complex visual tasks, since we're seeing these decrements in performance or selectivity for really simple cognitive tasks, then I would assume that as the task gets more complex, that we would see an even larger effect in some decrements.
All right, this next one is from Young Hee. Are there differences between effects of aerobic, example light cardio, versus anaerobic, such as weightlifting exercises, on working memory or any other higher order cognitive functions?
So, Yaki, I know you go to the gym. I know you're going to ask that question. So unfortunately, not a lot of people do research on anaerobic exercise. And so, I mean, the amount of literature that's on working memory during exercise is very sparse. And if you try to filter that down to anaerobic exercise, it's even sparser. I only know like of one paper, and I don't think it even showed effects. But in terms of how anaerobic exercise affects our cognitive abilities, it's been shown that It enhances information processing speed or just reaction time, and as well as long-term memory. I haven't seen much in terms of working memory, and its enhancements are much smaller than aerobic exercise. It's more infrequent than the ones that are shown by aerobic exercise, which is why a lot of people look at aerobic exercise instead of anaerobic exercise. I don't know if that answered your question. Can't get your facial feedback from that.
It's true. Next question is from Todd Kippen. Could this be related to tunnel vision experienced at high speeds? For instance, how race car drivers may process information to optimize navigation.
So it definitely could. We just recently, my project scientist presented a paper in our lab meeting showing changes in visual processing for peripheral locations, and it was showing that when you're in a physically active state, you're seeing this enhancement for items that are at a certain distance from fixation that are in the periphery. I forgot what angle it was, but items that are outside of that sort of angle or distance from fixation are shown to have no effect or like no enhancement of processing those items, or there's a decrement in processing. So in terms of like the tunnel vision, it definitely could be. I mean, also, so that's more of the peripheral, but the work showing the increase in the visual evoked response, all of those stimuli were presented at fixation, and we are seeing these enhancements for stimuli that are presented for fixation and for peripheral stimuli that are presented a certain location in the periphery. But for locations that are outside of those areas, it's possible that we could have some decrements. So that's my answer to like the tunnel vision. We have thought about that in terms of like when you're actually engaged in exercise, you're not really looking in the periphery too much because you want to focus more so on the fixation. Unless you're like a mouse that's trying to figure out how to get away from a bear. I've seen mice run for bears.
So I think that answered it because Todd says thank you. So from Evan, we have, how does exercise affect criterion shifting?
I know, I saw the question. You know, Evan, that's a great question, Evan. We don't know. I mean, no one's ever studied that. And you and I talked about that a little bit when you participated in the study. And you had very beautiful tuning functions. Thank you very much. But I mean, we saw some differences in response bias there, but they weren't. There wasn't moderate evidence for there being statistical difference. So it's possible. I mean, once you start to get fatigued or something like that, that you develop more of a liberal criteria, or that once you're seeing some enhancements in cognitive processes, that you might become more conservative. But honestly, I haven't read anything about criterion shifting during exercise. But if you want to collaborate, be more than willing to.
Awesome. That looks like that's the last of the questions for you, Jordan. Thank you.
Oh, yeah. Woo!
Well, we made it.
Great job, Jordan.
Thank you, guys.
I would now like to introduce Dr. Kyle Ratner, who will present the McClintock Award.
So hopefully everybody can see and hear me. Oh, wonderful. So first of all, congrats to all of today's speakers. I really enjoyed all of your talks. The reason I'm here though is to present the Charles G. McClintock Award. And although I never met Chuck, I wish I had. So Chuck was the first social psychologist at UCSB and built the program to international acclaim over many decades until his retirement in 1992. One of Chuck's close friends and colleagues, Dave Hamilton, notes, Chuck was, of course, a productive scholar and was well respected in the field, but he also cared a lot about education and was famous for his extensive and absolutely indecipherable scribbling in red ink on every paper handed to him by a graduate student. Beyond those qualities, though, and perhaps more importantly to those of us who knew him, Chuck was very people-oriented in his concern for others. And for these reasons, the award named after him is awarded to a social psych PhD student who's conducted sound research, but also put a high value on education and has had a genuine concern for others and for the functioning of the social program. So Youngie Hong, a graduate student in my lab, is this year's winner and exemplifies all of these attributes. Youngie has excelled in graduate courses, not only in social psychology, but also in cognitive and computational neuroscience and in statistics and applied probability. And it has been so fun for me to work with him over the past 5 years on a host of fMRI, EEG, and behavioral projects examining how group memberships influence face processing, and also how being perceived according to our group memberships influence neural processing of incentives. His work is particularly elegant in the methodological and theoretical sophistication. This is clear from a first author paper he recently submitted for a revise and resubmit at JPSP, fingers crossed. It challenges 50 years of understanding of classic minimal group effects in social psychology. That's the theoretical innovation. And it does so in part by applying representational similarity analysis from the fMRI literature to understand behavioral reverse correlation data. And that's an impressive methodological advance. He not only brings insight from cognitive neuroscience to inform social psychology, though, he also brings insight from social psychology to inform cognitive neuroscience. For instance, he's currently writing up a paper that uses multi-level modeling approach that's most popular in the behavioral social psychology literature to analyze ERP responses to in-group and out-group faces in a new way. And these are just two examples of a long list of projects that he has underway. In addition to his research efforts, he's an excellent teacher and department citizen. He has been the instructor of record for numerous summer courses and even taught undergrad stats during the academic year. And he's TA'd for Nancy's grad stats class multiple times. He's helped numerous fellow grad students with their analyses. He's mentored countless undergrad RAs, and he's always a cheerful and supportive presence in the lab and around campus. So congrats, Youngie, on this well-deserved recognition.
Thank you, Kyle.
Thank you, Dr. Ratner, and congratulations, Youngie. Great job, second years. You all did amazing, especially given the circumstances. This was a very unconventional Minicon, but you did amazing. Thank you to the third year cohort for all of your planning. This was led by Casey Mays. Thank you, Casey. You did such a great job. And thank you to everyone who attended. I'd like to now pass it on to Dr. Mike Miller, who will do the closing remarks.
Okay, can you hear me okay?
We can.
Okay, great. So, wow, that was just a great set of talks. Really, really good job. I'm still actually a bit stunned by how sharp Jordan looked there at the end. And hopefully Evan took notice of that. Anyway, the talks, as usual, were just great. And I think I can speak for all the faculty when I say that we are very proud of the work that you're doing. And, sometimes I just want to say in general that for all of our graduate students that, sometimes it takes, a crisis like this or extreme situation, like a campus shutdown or a coffee pot malfunction, to really help us sort of appreciate all the great work you are doing and how much you mean to us. And you really are the engines of our labs. And I just can't say enough how much all of us really appreciate that. So I'd like to thank Casey, Melissa, Rami, Leo, Michelle, Delancey, Laura, Vinny, Roxy, Diego, Julian, Madeline, Nicole, Carol, Debbie, Ashley, and Katie we're just putting on a spectacular event. I mean, this is a particular challenge this year. You didn't have the usual templates for this that we often have. And so your creativity and organizational skills have just been fabulous. And this is really terrific Minicon, and I greatly appreciate it. And just to show our appreciation, and I have Lynn and Callie working on this, we're gonna, and we quite haven't figured out how we're going to do this, but we're going to get you a pizza coupon for all the organizers and presenters. Just want to make clear this doesn't include Evan because we can't afford that, but we are going to find a way to get you that pizza you so well deserve. So thank you again for a great event, and I look forward to next year. Okay, goodbye.
All right, everyone, that concludes our Minicon. So I hope everyone has a great day. Thank you so much for joining us. Truly appreciate it. It was fun to put on. Very interesting, a lot of different hurdles to overcome that we didn't expect along the way, but I'm glad everybody was able to make it, and it seems like it went pretty well. So thank you all, and yeah, I hope to see you all soon.